So the first word for attendance is extra credit, since John is here early or online early. But if you're watching the video later, so not if you're online right now, but if you're watching the video later, this is just your regular first word for attendance. And the first word is soap, like something you would wash your hands with, soap. And of course, if you're watching the video, we've got about 7 minutes before we get started, so you can now skip ahead about 7 minutes. right, we've got a minute left. I guess I'll do attendance. Would be easy, but we're recording, so I also just want to get myself, as usual, recorded saying who's not here, as opposed to not so much as who is here, for obvious reasons. Bangura is not here. Case Bolt is not here. Tristan is not here. Covington Griggs is not here. DeVere is not here. I was here. And if you continue to be the only person that's here, instead of getting, I'll just double your points, put it that way. Anyway, Hughes is not here. Lundy is not here. Little is not here. Mankins is not here. Mathis is not here. Rains is not here. Reed is not here. Elise is not here. Thompson is not here. Yindui is not here. And Elkins is not here. In other words, the only one who is physically here is Morgan. Like I said, that'll be double the points. Even though I'm not officially doing announcements yet, you can see, like I said, on Monday, starting today, I'm doubling the extra credit points for attending in person because so much of the problems that people are having is because they're just not attending or paying attention. So starting today, you get 4 points for attending in person. And Morgan, like I was saying, if you happen to be the only one for the rest of the day, then I'm going to double that to 8 points of extra credit. Anyway, it's 8 o'clock. Let me jump into this. Does anybody have any questions while I get this PowerPoint pulled up? All right, so let me back it up. As usual, I don't want to start with the announcements. So today's going to be a little bit strange because we're going to barely do anything from Chapter 8 because we're almost done with Chapter 8. We're going to quickly finish it, and then we'll jump into the next chapter. But this is where we left off. We talked about sex-linked genes, and as I mentioned on Monday, if I have time, I hope to come up with a new worksheet so you guys get practice doing these problems where I'm like, all right, you know, in the question on the exam, I'll say, here's the story with this thing, right? This is how this sex-linked gene works, right? Is it recessive or is it dominant? Is it on the X chromosome or is it on the Y chromosome? Right? I'll explain that in the question. And then I'll say, if this person mates with this person, what are the chances of their offspring being that? Like using color blindness as an example, and this is the last slide we looked at on Wednesday. This is a great example. I would explain how color blindness works, right? So I would explain to you that this is a recessive disorder, meaning 100% of your alleles have to have the recessive allele, you know, to get the recessive disorder. And it's on the X chromosome, right? So I would tell you those two things. And then on the exam, I would say, what would happen if a, let's see, unaffected or normal female, which is right here on the left, mated with a colorblind male? And then you would do a Punnett square, and you know, and then I might say, what are the chances of their kids? What are the chances of them having a male that is normal? And you can see here, the chances of them having a male that was normal would be 50%, right? Or I might ask, what are the chances of them having a female that is a carrier? And you can see here, the chances of that would be 50%. Or I could get tricky and say something like, what are the chances of them having a female, which is XX, that is colorblind, which would be little n, right? And you can see here, none of those meet that criteria. So the answer to that would be 0%. So those are the kind of questions you might get on the exam. And again, I would give you all the pertinent information. It's not like you need to memorize the fact that color blindness, and using this as an example, is a recessive disorder, and it's on the X chromosome, right? I will give you all that. But you would need to be able, from once I give you that information, to understand terms like unaffected individual or normal, right? You would need to understand what a carrier is, and you would need to understand, obviously, if somebody, in this case, they have the condition as colorblind, but obviously if they use something different, on the exam it would be different than colorblind. For attendance words, we're going to do the same thing as usual. I'm going to ask you a question based on some information that I just gave you. The last word for attendance, if you will, is to ask me a question about the material presented. So feel free to ask me anything you want. Last Monday, somebody asked me a question about independent work, which is great because I want to answer your questions about that kind of stuff. But you know, to get the credit, I need you to ask me questions about the material. And of course, if you're online right now, if you're watching the video as opposed to online, as usual, I need a screenshot of when I say it. So a screenshot in addition to you typing out the words. Anyway, the first word, if you will, for attendance, the first number for attendance, and there's 3 options because I've given you all 3 options. given this mating up here of an unaffected individual female with a colorblind male, what are the chances that those two would have a female who is colorblind? And I gave 3 examples, and they're both written in there with my handwriting. So one of those 3 is the answer. One of those numbers, one of those percentages is the answer. And of course, Morgan, since you're here, you don't have to send these words in, but if you do, it's extra credit. Anyway, that's the first word for attendance. Again, I'll remind you before I move forward, this is just an example. So when you're studying, like if you're looking at the PowerPoints, because this picture is not from your textbook, it's from the old textbook, so you can only find it in the PowerPoints. If you're studying this PowerPoint, don't try to memorize, all right, if this person mates with this person, then this is what's going to happen, right? Think about in your brain how that Punnett square is working out. And in a sense, you almost want to think about like math, right? You want to figure out like, all right, how to use this, how to work with this problem to come up with your answer. Yeah, so let me give you another example from color blindness. Again, this is just the one example where you're just using color blindness. So again, I would give you all the pertinent information in the exam, but then I would ask you a question like, all right, what would happen if you mated a carrier female? Because you can see here, she's homozygous, or excuse me, heterozygous. Right, so she's got one normal allele and one recessive allele, therefore she's a carrier. So I would say, what would happen if you mated a carrier female with a normal male? What are the chances of them having a completely normal female? Right, and that would be 25%. What are the chances of them having a completely normal male? That would be 25%. What are the chances of them having a complete carrier female? 25%. What are the chances of them having a colorblind male? 25%. Before I move forward, and I should have mentioned this last slide, I just want to point this out. So far, when we've been talking about dominant versus recessive, I've always said that in order to have the recessive disorder or phenotype, because when you think about it, a disorder is a phenotype. In order to have the recessive phenotype, you have to have 2 recessive alleles, right? And that has been true up to this point. Now I want to rephrase everything I've ever said. So now instead of saying you have to have 2 recessive alleles, the better way to say that, the more accurate way of saying it is to have the recessive phenotype or disorder, that you have to have 100% of your alleles have to be recessive. And I point that out, I make this distinction because in this case, when we're talking about color blindness, a male, since you only have 1 X chromosome, you're only going to have that 1 recessive allele. But you're not going to have any dominant alleles. So even though you only have that 1 recessive allele, the point here is 100% of your alleles are recessive. Does that make sense? So keep that in mind. And we're changing things up just a little bit. Now, instead of saying to have a recessive disorder or recessive phenotype, you have to have 2 recessive alleles. No. Now the better way of saying it is 100% of your alleles, whether it be 1 or 2 or even more, which we haven't talked about, they have to be recessive. Yet another way of saying it too is this. In order to have the recessive allele, the recessive phenotype or disorder, you cannot have any dominant alleles, right? That's another way of looking at it. All these different ways of saying the same thing. I just don't want you to get stuck on like the definitions and the exact way I said things or the exact way things are said in the textbook. I want you to think and don't stick with words, right? Think about how things work. Anyway, again, this is just an example. Any questions about that example before I give you, I think, just like one last example? All right, so again, on the exam, the question might say something, all right, if you mated, if 2 people mated and there was a carrier female and a colorblind male, which is what we have here, what are the chances of them having a carrier female? And they go, oh, that's 25%. What are the chances of them having a normal male? 25%. What are the chances of them having, well, we'll change it up a little bit. What are the chances of them having a colorblind kid? Right, so just forget male or female. I could ask that too. What are the chances of them having a colorblind kid? 50%. Right, or I could get specific. What are the chances of them having a colorblind female? 25%. What are the chances of them having a colorblind male? 25%. You guys good with that? That makes sense? Again, the idea here is a Punnett square. And again, I've already created 2 documents, worksheets, where you can practice doing Punnett squares on simple complete dominance. I can see the dominant or it's not, right? Like chin dimples and all that stuff we first did. Then the second worksheet starts diving into things where, again, you're doing Punnett squares, but then you start dealing with things like incomplete dominance and codominance. And then hopefully today, I'll make a third worksheet where you get some practice doing this here, which is, sex linked. And really, so far, and I don't have too much time to talk about it, but now that I think about it, the only example I actually gave you when I worked Punnett squares, which is what we just finished, was a recessive disorder on the X chromosome, right? So we haven't talked about dominant disorders on the X chromosome. We haven't talked about recessive disorders on the Y chromosome. We haven't talked about dominant disorders on the Y chromosome. So again, I hope to make a worksheet so you can get some practice and extra credit, right? It's all worth extra credit too. It's also a good independent work topic. So here I've only talked about color blindness. This will be so easy, depending on, if you don't like to write, this could be a very easy independent work topic. You can just look up other stuff and not even get really into it. You can be like, all right, so this disorder is a dominant disorder on the Y chromosome. And just name it. Here's the name of it. Here's my source. Here's where I found it. This disorder is a recessive disorder on the Y chromosome. Give me the name, give me the source, right? You could just list a few things. Because like I said, I've only given you this one example. Well, I'm about to give you one more example. But other than the examples I'm giving you, you can look that up for independent work. Anyway, any questions about this example? Or color blindness in general? Okay, and just as a reminder, when you're studying, you do not need to memorize anything about color blindness. Any questions on the exam, when I say this thing mates with this thing, what are the chances of that thing being an offspring, I will give you this pertinent information, right? I would tell you this is, in this case, I would say this is a recessive disorder found on the X chromosome. I'll give you all that pertinent information. Let me give you another example that I'm not going to ask you about on the exam, so I'm actually going to go through it pretty quickly. Hemophilia is a big example. This is basically you're a free bleeder. It's a sex-linked recessive disorder with a well-documented history. The reason it has a well-documented history is because there was actually a lot of royal people in the royal families in early Europe that had it. You can look into it if you want to. I'm not going to spend time talking about it because I'm not going to ask you any questions about it. Notice too, I didn't even tell you what kind of disorder it is. So again, for independent work, you can look that up. Hemophilia. Is it X chromosome? Is it Y chromosome? Is it dominant or is it recessive? You can look that up. But anyway, yeah, if you're into history, this is actually pretty interesting. And not just, this is just one example, but if you're into history, that's something you could look up for independent work. Like, when has biology changed history? This is kind of one of those times. I don't know how much you know about the Russian Revolution, but with Alexis having hemophilia, this is back in the day, right? It's not like they had like great medicine. So they brought in this guy named Rasputin, who's kind of supposed to help him. And he was also, though, instead of just helping Alexis, Rasputin was also influencing the royal family. And that really pissed off the people in Russia, hence the Russian Revolution. Of course, I really simplified it. It's a little bit deeper than that. And I'm not getting political, but just as a good analogy, you could almost think of like, you've got Elon Musk, who's like all tied in with Trump. He's not actually part of the government, wasn't elected, but he's obviously influencing him. Same kind of thing here, right? And again, I'm not getting political, so I'm not saying that as if it was a good thing or a bad thing. I don't care what your opinion is about Trump, and I hope you don't care about mine either. But anyway, look that up for independent work if you want. This is another thing left over from the last textbook, so it's not in your textbook, so I'm not going to ask you about it, but if you download the PowerPoint, you can read about it. It is slightly interesting. Yeah, anyway. So before we jump into the next chapter, let's quickly do announcements and reminders. Most of these are just reminders, actually. Again, lab is in person today. Despite the fact that it's virtual, we're going to meet. So if you have done it, you'll know if you don't have to come, because I would have already told you. I think 3 people turned in a lab overnight. I haven't had a chance to grade it, but I looked it over and saw that it was good enough to where you don't have to come in. So I left a comment on your lab and said you don't have to come in. But the rest of you should come in. Make sure you bring your computer, because again, it's virtual. We're all just going to be sitting here doing our own work, and I guess that's it. And I'll be here, or the TA will be here to help you. Anyway, any questions about today's lab? Okay, exam 2. Again, the retake is still open. Again, when you're done with it, you need to tell me. Your grade will not automatically change. You have to email me and say, hey, I'm satisfied with my retake. I've taken it enough. This last time I like the grade, so you can go in and average the first grade and the last grade. So make sure you let me know. Any questions about the exam 2 retake? Again, open book, not timed. You can take it as many times as you want. Your grade will never go down. You have nothing to lose. Not one person took me up on that on exam 1. Hoping somebody will for exam 2. Last day to drop. So I've been saying this for a while. I've been saying that it was this month. And as promised, I finally looked it up. So it's this Friday. So some of you should really, not some of you, because you're here. Probably I'm preaching to the choir, as a matter of fact. Probably anybody who needs to drop is not here. They're not online, and they're not watching the videos. But just in case, you should probably, some of you should really consider dropping this course. And if you decide to stay, please let me help you. Some of you really have some catching up to do, and I can help you do it. Independent work is still not added to WVUSU Online. I know I said I was going to do that after midterms. I've committed to doing it today. Since Friday is the last day to drop, I want you to see what your real grade is. So I'm hoping to get that done today. Again, like I said, on Monday, I'm reminding you starting today, you get double extra credit for attending in person. Speaking of which, if you came in late today, make sure you email me and tell me you came in late. Instead of getting 2 points for coming in person from now on, you get 4 points of extra credit. And remember, that all gets added at the end of the semester, so that can really help you. And then once again, like I keep saying, the Monday after Easter and the Friday before Easter, office hours and lecture will be 100% online. We will not be in person. Any questions about that? All right, you guys, nice and easy. Really quickly, let's do a quick review of this chapter before we move on to the next chapter. We can ignore that. The law of segregation, you definitely need to know the law of segregation. Remember, the law of segregation, depending on the context of how you're looking at it, let's look at it in 2 different ways. We can do it both with this picture. The law of segregation says first we'll look at it as genes, right? For any gene, that has 2 alleles, when you produce gametes, when you go through meiosis to produce sperm or egg, they segregate, right? So the allele that you got from your mom goes one way, and the gene that you got from your dad goes the other. You guys got that, right? That's the law of segregation. Those alleles segregate from each other. Saying the same thing in the context of chromosomes instead of genes, the law of segregation states for each pair of homologous chromosomes, you got one from your mom and one from your dad. When you go through meiosis and you produce your sperm and your egg, those chromosomes segregate. Those homologous chromosomes segregate. So the one you got from your mom went one way into one cell, and the one you got from your dad went another way into the other cell. Make sense? That's the law of segregation. So this is kind of a question that you might get to on the exam, where I might say, again, I've already mentioned this before, so I'm just saying it again. I might say, this thing was mated with this thing, here were the results, or what are the chances of this being the results? Or, this is a great picture, I'm glad I saw this to remind me. I might say, this thing mated with this thing, these were the results. What's the genotype of this thing, right? So that's something you might know. Let's see. So for example, if, again, in the question on the exam, if I were to give this as a question, I would say, a purple flower plant mated with a white flower plant. The results were all purple flowers. What was the genotype of the purple flower plant? Right? I might give you a question like that, and that's enough information. You already know, first of all, that the white flower plant, well, I would tell you, I guess, a purebred white flower plant, right? So I would say those things mix. If the F2 generation, right, if these cross and it's 100% purple flowers, then clearly this thing must be dominant, right? Because that must mean these are all heterozygotes. All these are heterozygotes. But if I said, again, a purple flower plant mated with a purebred white flower plant, and the offspring were about, half and half white and purple, what is the genotype of the purple flower? And then you could know, all right, for sure, that purple flower has to be heterozygous. Otherwise, there would be absolutely no way, other than mutations, which we haven't talked about yet, there'd be no way that there could be any white flowers, right? Because those white flowers had to have a recessive allele somewhere, right? So it would have to come from right there. So you might get a question like that. And again, that's an example of complete dominance. Here's a quick example of incomplete dominance. So to remind you, incomplete dominance is when you're blending the phenotypes, and this is just a classic example. Red and white makes pink, right? So the heterozygous and incomplete dominance is always a blending of the 2 phenotypes. Pleiotropy, really quick as a reminder. Pleiotropy is when you have a single gene, but it doesn't control a single character. It's not like one gene for the chin dimple and one gene for freckles. In this case, it's a single gene, but it controls multiple traits. Sickle cell was the example. I can almost guarantee you sickle cell will not be the example on the exam, because I don't want you to memorize the fact that sickle cell is pleiotropy. I want you to be able to recognize something completely new and say, oh yeah, that's an example of pleiotropy. So might make up something, a completely made up disorder. But the point is, if in the description of that disorder I say there's a single gene and I list multiple traits, then you would know it's pleiotropy. Of course, the opposite of that would be polygenic inheritance. So in this case, you would have multiple genes that affect a single trait. And again, height was the example we used for that. And then Monday, I said that would be a great topic for independent work, right? So I gave you that one example. What are some other examples? Also, same thing with the last one we just talked about. Pleiotropy. The one example I gave you was sickle cell. What are some other examples? Again, this is how I've seen some people miss this. I don't remember if this was you guys or the one on the in-person class, but I saw a lot of mistakes on this. So to remind you, we have 46 chromosomes, right? We have 23 pairs of chromosomes. 44 of those chromosomes, or 22 pairs of those chromosomes, are what we call somatic, right? And then the sex cells obviously would not be those. Sex cells would be your egg or your sperm, aka gametes. Really quickly, this is good, you might want to study this. If I remember correctly, this is not in your textbook, this is only on the PowerPoint, but it's a nice little table where you can visualize all these things when it comes to sex-linked traits, right? So female versus male, you can read it all. But anyway, that's a good table. Also, that's another good way to study. I just thought of that. If you want, maybe that's a good, depending on how your brain works, different topics, that might be a good way for you to study it. It's like, all right, let me make a table. It's like complete dominance, incomplete dominance, codominance, right? Make a table and have little descriptions so you can just have it that one table and study it that way. Depends on how your brain works. Some people prefer to read and have it in paragraph style. Some people prefer pictures. Some people prefer tables. So do what you need to do. Again, just really quickly, you really should be familiar with Punnett squares. Again, I have those 2 documents that you can do, those 2 worksheets you can do for extra credit, hopefully 3 soon. And this is just an example, right? So this is an example if you mix 2 heterozygote people. And again, I might ask you a question like that, right? All right, you have 2 heterozygotes that can both hear, but they're carriers. What are the chances of them having a deaf child, right? You would do a Punnett square and say, oh yeah, 25%. What are the chances of them having a carrier child? 50%. What are the chances of them having a homozygous dominant child? 25%, right? And then I could get even crazier, if you will. And again, I've done this on this worksheet, so you can have practice. I might say, what are the chances of having a deaf? So you already figured that out, right? 25%. What are the chances of them having a deaf boy, right? So the chances of them being deaf, we already see is 1/4. The chances of it being a boy or a girl is 1 and 2. So therefore, the chances of them having a deaf girl or boy would be 1 and 8, which, just so you know, is about 12.5%. So you can have a calculator when we do exam 3, just so you know. You will have some very basic math like that. Matter of fact, before then, maybe, I don't want to say that. Okay, so that's it for this chapter. I'm going to jump into the next chapter. Does anybody have any questions while I'm doing that? Okay. So bear with me. That's why questions would be good, to fill the silence. Almost feels like a radio show in the mornings. Like, if you're doing a talk show on the radio, you want to have constantly talking or something. Not a lot of silence. Sorry, I should have had this pulled up earlier. Actually, I might. Let me double check and make sure I don't have chapter 9 already downloaded. So, since you guys have already, some of you have already done this, the lab for this week, and hopefully all of you have at least done the pre-lab, and you know what that lab is about. Therefore, hopefully, you know what this chapter is about. Anybody want to guess, based on what I just said, what this chapter is about? Hint, what lab is due today, or Oh yeah, I guess that is true too. What kind of lab? What are we doing in lab this week? What is this week's lab about? That's your hint. Nobody? You guys just being shy? It's okay.
DNA.
Yeah, DNA. Great. Perfect. Yes. This chapter is all about DNA. And remember, you've already been introduced to DNA. Back when we did that biological molecules section, I said, I'm introducing you to DNA right now. This is just an introduction. We're just scratching the surface. Later, you're going to learn more about it. And this is now later. So now we're going to dive deeper into it. It's downloading right now. Another time you've been introduced to it slightly was when we talked about the S phase of interphase, right? So you know that DNA duplication happens in S phase of interphase. And we even talked about the process a little bit, right? So you understand this concept of having that single chromosome, and then it's doubled, and then you have sister chromatids, and then those sister chromatids are separated, and now they're full-blown chromosomes again. So that's where we're at. Now we're just going to dig a little bit deeper. So here we go. So technically, this is molecular biology. Yes, we will be learning about DNA, but also we're going to branch out a little bit and talk about some of the, I don't want to say the technology, but some of the stuff involved with DNA. Let's put it that way. So, again, this is from the old textbook. I like the old textbook for some reasons, and this is one of those reasons. I like the pictures at the beginning of the chapter. Obviously, this is a person holding a vial. And the question is, why does molecular biology matter? Well, that picture is one example. And the old textbook said, okay, all life on Earth shares a universal genetic code. So your DNA can be used to genetically modify bacteria and vice versa. And that's a big one. We're not going to talk a lot about it, but for your everyday life, this is actually a really big deal. I've seen a lot of misinformation about the COVID-19 vaccine. So in this building, we do stuff like this, right? So in this building, if we want to study a gene, we do this a lot, we'll take the gene from whatever we're studying, usually in this building, it's usually plants. But anyway, we'll take that gene that we're interested in, we'll cut it out of the genome of the plant, if that's the example, and we'll insert it into, we'll use a virus sometimes to insert it into a bacteria. Why do we do that? Well, bacteria are really good at reproducing, right? So therefore, every time that bacteria reproduces, it's going to reproduce that gene, that section of DNA that we're looking at. That's just something we do all the time. And just as an example, not that you need to know this, I saw this thing about some study that they did for COVID-19. They took the genetic information that was in the COVID-19 mRNA vaccine, right? They took that genetic information, they inserted it into bacteria to study it, like the way we do all the time in this building too, and all over the place. And they just did a study. I forgot the point of the study. But the way that was twisted, the way the people twisted it, and I'm not giving you an opinion on how safe or effective the vaccine is, because I hate giving opinions, but this was a really stupid argument. So somebody read that, somebody who doesn't understand biology, and they read that and like, oh my God. So the genetic information from the COVID-19 vaccine can genetically modify an organism, because I mean that's what we did, right? That was the process. So I'm just saying this as far as I know, and I haven't read any studies. I'm just using this as an example. The mRNA vaccine, and you're going to learn more about mRNA in this chapter. The mRNA vaccine cannot mess up your DNA. As you're going to learn, it goes from DNA to RNA to protein. So again, these idiots, well, let me take that back. These underinformed people or liars, whichever one they were, I don't know, were all up in arms because someone took the genetic information from there and deliberately put it into bacteria, which you can do for anything, right? I can take a swab from your cheek, I can pick out a gene from your DNA, and I can insert it into a bacteria in this building. For what reason, I don't know, but I could. So it's not that uncommon. Anyway, my point is, I guess, since this is the introduction to this chapter, this is a very important chapter these days for understanding things. Another example, actually, this is kind of a bad example. Your book example for this was mad cow disease, right? So you can learn more about mad cow disease in this chapter, but that's really not that important. If I were using that picture as an example, I would talk about GMOs. So you can learn a little bit more about GMOs in this chapter, because that's all about molecular biology. And then, of course, like I mentioned earlier, this is sickle cell, and I said earlier when I first introduced you to it, I said, all right, this sickle cell is going to come back. We're going to use it in other examples. And this sickle cell is a great example of mutations. And you're going about mutations for this chapter. Anyway, your next word for attendance is to tell me what is wrong, because I just used the word. What's up with this cell? What do we call that? I just said it. Don't worry about spelling it wrong, but that's an example we used in the last chapter. That's an example we're going to use in this chapter for to talk about mutations. So what do you call that disorder, or how would you call that? Describe the shape of that cell? I just said it. And again, don't worry about spelling. Any questions about this slide? All right, so again, these PowerPoints are based on the old textbook. And when I say old, I don't mean just old as in like we're not using it anymore, but old as in it was published a long time ago. So back in 2016, which is when I think that old textbook was published, a really up-and-coming thing that was scaring everybody was Zika, the Zika virus that makes like babies born with small heads. So you can read about that if you want. I'm not going to ask you any questions about it. Again, the reason but I do like it. What I love about that old textbook was before they started teaching you all this stuff about biology, like, well, the mRNA, does this and that, and the really boring stuff. They started off with something like, here's something that's really going on in real life. So anyway, read about it if you want. Obviously today, if they were to publish this, I bet if I were to look at the newest edition of that, this chapter for this introduction will probably, I would assume, be about COVID-19, right? That's one of the newer things. Or maybe monkeypox, or maybe bird flu, who knows? Anyway, that being said, again, this PowerPoint is based off of the old textbook. So if you're following along in your textbook, it might be a little bit out of order. Some of the examples might be a little bit different. Some of the pictures might be a little bit different, but that's okay. The point here, I just don't want you, again, if you're following along in your book, I don't want you to get confused by, whoa, what? How did we get here? I thought we were just talking about this, right? So it is in a different order, slightly. And this chapter is broken down into 3 main points. First, we're going to talk about DNA structure and replication. A lot of that will be a review, because we've already talked about a lot of this. Next, this is going to be, in my opinion, historically, this is the hardest part. The harder part for students is this middle bullet point, when we talk about how we actually go from DNA to RNA to protein. In my opinion, it's actually one of the more interesting parts. And then finally, at the end, knowing all this stuff that we know about DNA and how we go from DNA to RNA to protein. Once you're familiar with that, then we can start talking about stuff like viruses and other non-cellular infectious agents. So here we go. Let's talk about this. So again, this old textbook, as you already know, I mentioned this before. A lot of times when they present something, they present it as a story, which I really like. So it's like, at first we knew this, and then we knew that, and then so-and-so figured this out. So I'm going to present it that way because, again, in my opinion, that's more interesting. But just before I do, I just want to make sure you know I'm not going to ask you any of these dates. So I'm giving it to you the way it's presented in the old textbook. I'm giving you these dates, but I'm not going to ask the dates. You might need to know chronologically what happened, but probably not that either. So that being said, let's jump into it. By the late 1800s, scientists knew that DNA was a part of the cells, right? So scientists were able to look inside of cells, and do different chemical reactions and say, all right, cells have this, and cells have that, and cells have this other thing. They didn't necessarily know what they did, but they knew some of these things were there, including DNA. So for independent work, you can write that up. That's pretty vague, right? We knew that DNA was part of the cells by the late 1800s. Well, first of all, how did we know that? And exactly what date did that happen? And who figured it out? And how did they figure out? Was it a chemical thing where they did chemical reactions like, oh, well, we found this chemical? Or was it like a visual thing where they say, oh, look at those chromosomes. Let's call that whatever they called it back then. I actually don't even remember. So that's a good independent work topic for that first bullet point. Then in the 1930s, they figured out that there was one kind of molecule that was the basis of inheritance. They didn't know which molecule, but they knew somewhere in that cell, this is how this is happening. Everything Gregor Mendel figured out, because that was way before the 1930s, someone figured out, they said, all right, everything that Gregor Mendel has figured out, that is by all those ratios and all the probability, that's all from some kind of molecule inside the cell. They didn't know what kind, but they knew it was a molecule. And again, a great independent work topic in the 30s, right? So exactly when in the 30s? Who figured it out? How did they figure it out? what kind of molecule did they think it was, right? So again, they didn't know it was DNA. But think about it, it's not like they just went from not knowing at all to going, it must be DNA. No, surely there were some people who had some hypotheses that they tested that were incorrect, right? So you can look some of that up. Like, John Smith looked it up and he thought it was, he thought carbohydrates were the material. And these are the experiments that he did, and he was wrong, right? So you can look all that up for independent work. So then in the 1940s, the new discoveries, again, at this point, they didn't know about DNA. So they found out that chromosomes, they're learning more about chromosomes. They learned that chromosomes, as you already know from previous chapters, are made up of two components, DNA and protein. You've already seen this picture, right? So the DNA wraps around those little proteins called histones. And for there's 8 little histones for each nucleosome, right? So when you wrap the protein, the DNA around 8 little histones, we call it a nucleosome. All this stuff you already know. But they figured that out in the 1940s. And once again, for independent work, who figured it out? How did they figure it out? Exactly when did they figure it out? Any questions about this slide? There's still another decade on the next slide, so we're not done talking about this timeline. All right. Excuse me. In the 1950s, we discovered that DNA is the molecule that acts as the hereditary material. So that's when it happened. In the 1950s, finally they figured it out. And think about that, that's a huge deal. We take it for granted because this is something that you're going to be tested on. Good grief, why do I have to get tested on this when I'm a business major, right? But that's a really big deal, especially back then. So that they are, again, Mendel already figured out all that stuff that I taught you about with the pea plants. And then it took them this long, and then finally they figured out this is the molecule that's responsible for it. And that ushered in the era of what we call molecular biology, which is the study of heredity at the molecular level. So it's not like what Mendel did, not like what a lot of your test questions are going to be like, all right, we mix this thing, and what are the chances of the offspring being that, right? That's just regular genetics. Here, we're really looking at it at the molecular level. So when we say we cross this thing with this thing, we're talking about like, all right, what are the molecules doing when this happens? And that's what this chapter is about. Any questions about that slide? All right, so this is again a continuation. Once we figured out the DNA was the hereditary material, then there was like this big push to figure out the structure. As hopefully you remember from chapter 1, and I've been bringing it up since then occasionally, more so when we were talking about molecules and chemistry. But remember, structure and function, right? So the structure of a molecule or the structure of an atom, whatever, usually molecule, the structure of something really tells a lot about its function. It tells what it does. So because of that, even though they, once they figured out the DNA, that was the molecule responsible for heredity, they didn't know how it worked. They just knew it was DNA that did it. And again, for independent work, how did they figure that out? Who figured it out? Once they figured that out, they're like, all right, we need to figure out the structure. If we can figure out the structure, then we can figure out how it works. So what they did know, even though they didn't know the three-dimensional arrangement of it, they did know all of its atoms, right? So if you, for example, if you think of a particular nucleotide, right, they knew all the carbons that are in that sugar and all the all the molecules, the atoms that are in the base and the phosphate. They knew all the atoms that were present. They just didn't know how they were put together. Like how do I put that different? They knew how they were bonded together, but they didn't know the actual structure. And again, independent work. How did they know this? Who figured this out? Anyway, again, they did not know the three-dimensional structure. They didn't have that, therefore they didn't quite understand the function. So therefore, that when I say the function, what I mean is this. Generally, and I'm probably not going to test you on this. So far, most of this has been leading up to what you need to know for the exam. So far, this is mostly just an introduction. But anyway, its unique properties are basically boiled down to these three things. DNA can store genetic information. And at this point, in the 50s, they knew that DNA could store genetic information. They just didn't know how. They knew that DNA can copy genetic information. That's a unique property. They didn't know how. And they knew they could get passed down from generation to generation. But again, without knowing the structure, they didn't know how. They didn't know how it had these unique properties. Any questions about this slide before we finally jump into the more important stuff? This is, again, most of this has all been an introduction to what we're about to jump into, which is DNA and RNA structure. Again, a lot of this should be a review for you. DNA and RNA are what we call nucleic acids, hopefully as you remember from chapter 2. They are polymers of certain kinds of monomers. So remember, the monomers are called the nucleotides. And to remind you, a polymer is a really big molecule that's made up of repeating monomers, right? And the analogy I used was a train, right? So a train would be a polymer, and each individual train car would be the monomer. And granted, some of those train cars might be different. Like one might carry people, one might carry coal, one might carry cargo, right? But they're all together, even though they're different, they're still individual little train cars, and you put them together to form a train. Same thing here, right? So the polymer is the nucleic acid. It's a polynucleotide, and they're made up of these individual monomers, which are nucleotides. Now, granted, the nucleotides are different. There's 4 different types of nucleotides, right? There's an A, a C, a T, and a G, if we're talking about DNA. And there's an A, a C, a U, and a G, if we're talking about RNA. So again, there's those 4 different types of nucleotides, depending on what kind of polynucleotide we're talking about. And that's what makes up the polynucleotide of DNA or RNA. And again, just to remind you, to give you real life examples instead of the train example. So for example, proteins. Remember, proteins are made up of amino acids, right? So the amino acid is the monomer. There's 20 different amino acids, but you put them together, you link them together in different combinations, and that's how you get proteins. Glucose is a good example, right? That's a carbohydrate. Excuse me, not glucose, sorry, starch. Starch is just a bunch of glucose monomers strung together. Right? So again, polymer monomer. This is an old concept. It's coming back. Any questions about this slide? All right, here's a picture of DNA and RNA structure. Excuse me. Well, no, excuse me. This is a picture of DNA. So here you have this drawing of the double helix on the left. In the middle, we've kind of straightened it out. So instead of seeing this 3-dimensional structure, we're just now more focused on the different parts. Right? So here's a monomer. Here's a monomer. Here's a monomer. Here's a monomer. Here's the monomer. Remember, the monomers are nucleotides, so each one of those is its own individual nucleotide. This nucleotide's an A, this one's a C, this one's a T, this one's a G, this one's a G. And then this picture is the same thing as this picture, is just drawn a different way. So here we're just saying, this is really basic, right? Here's the monomer. Here we're giving you more of like the shape of the molecules, right? Here's your phosphate group, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And hopefully you'll remember, and I'm sure I'm going to have this up on the slide in the PowerPoint in the next few slides. Remember, the sugar and the phosphate, those are all the same. Whether you're dealing with an A, a T, or a C, or a G, the sugar and the phosphate is the same. The only thing that makes an A, a T, a C, or a G unique from themselves, from each other, is the base itself, right? So this base is an A, this base is a C, this base is a T, that one's a G, that one's a G. So it's the base that makes it unique. And again, as another reminder, an example of that would be the amino acids, right? All the amino acids, right? That was a central carbon with 4 parts around it. And 3 of those parts were exactly the same, no matter what amino acid you looked at. The only part that was different in the amino acid was that side chain, right? It was the side chain that defined it. It was the side chain that determined which of the 20 amino acids it was. For this DNA, it's that nitrogenous base that's different. That's the thing that defines it and says which of the 4 nucleotides it is. Any questions so far? All right. Even though I've already said this, now we can see it in writing. Oops, I went the wrong way. There we go. The nucleotides, again, I mentioned this before, I'm mentioning it again. They are joined by covalent bonds, right? So if we're talking about a single strand of DNA, which we are currently, that nucleotide is covalently bonded to that nucleotide, which is covalently bonded to that nucleotide, which is covalent, so on and so forth, right? Let me ask you this. Let's see if anybody remembers this from the chapter 2. And I said this was going to come back, so here it is. I said the covalent bonds were either the strongest or the weakest of the 2 bonds, and I said that's going to come back and be important later in the semester. Does anybody remember which one it is? Are covalent bonds the strongest or the weakest of the bonds? And I don't blame you for not remembering. It was a long time ago. We've talked about a lot of different things since then. Haven't talked about bonds in a while. Covalent bonds are the strongest bond. Again, that will be coming back. That'll be important later in the semester. Anyway, when we're talking about these covalent bonds, again, this thing bonded to that thing, so on and so forth. What we call that a sugar phosphate backbone. I might ask you that. I'll put a question on that, question mark. I might ask you what a sugar phosphate backbone is. I can almost guarantee you that word is going to be used in a question. So for that reason, you need to know what it means. So they call it a sugar phosphate backbone because that's how the nucleotides are joined, right? The sugar of one DNA nucleotide is bonded covalently to the phosphate of another DNA nucleotide, or RNA, if that's what we're talking about. In this case, we just happen to be talking about DNA, but it would be the same if it was an RNA, right? Sugar phosphate backbone. Any questions so far? Okay. So, as you already know from previous discussions back in chapter 2, there are 3 parts, right? So if we're talking about nucleotide, which again is the monomer, there's 3 parts. And as a reminder, this is the example I used before for comparing and contrasting. If we're talking about proteins, remember the monomer of the protein is the amino acid. There's 20 different types of them. There's 4 parts of an amino acid, right? And again, the important part was the side chain, because that's what defined it. So the 3 parts of the nucleotide, there's 3 parts, not 4. There's the nitrogenous base, the sugar, and the phosphate group. You already know that. We've already said that, but let's talk about it some more. The nitrogenous base projects from the backbone. I'm not necessarily going to ask that, but you should remember that. So, again, if you're just thinking of this zigzaggy backbone, sugar phosphate backbone of a DNA molecule, again, it's the base. I'm just going to draw a B there for base. It's the base that sticks out. Right? Everything else, the sugar and the phosphate are bonding to each other, right? So it's the base that's sticking out. And that's not trivial. That will be important later. Because remember, in this case, I'm showing you DNA. Remember, that's only a single strand of DNA. Remember, DNA is double-stranded. So that's why this is going to be important later. Those nitrogenous bases are not covalently bonded, in the backbone. They stick out, right? That will be important later. They have a ring that's made up of nitrogen and carbon and other various chemical groups. I'm not going to ask you that, so I'm going to put it next to this. They are basic, not neutral, not acidic. Hence the name. I'm not going to ask you that. And again, there are 4 varieties, and that was on the previous slide. So if we're talking about DNA, the 4 varieties are A, T, C, or G. If we're talking about RNA, it would be A, U, C, or G. So yes, for the nitrogenous base, what you need to know for the exam is, first of all, that's one of the 3 parts. And second of all, it projects out from the backbone, right? It's not part of that covalent bond between the sugar and the phosphate. And there are 4 different types. More importantly, if I'm going to word this differently, it is the nitrogenous base, like I said previously, that defines what nucleotide it is, right? Because the sugar and the phosphate, whether you're looking at an A or T or C or G, they're all the same. The sugar and the phosphate part is the same. It's the nitrogenous base that defines which nucleotide we're looking at. So any questions about the nitrogenous base?
So is there a bond between the 2 strands of the nitrogenous base of the DNA?
Yes, and I'll come to that, yeah.
The acidic guanine and
Yep, yeah. And great question, and I'll come to that. So for now, before we even I love that you're looking ahead. You're talking about the double strand, right? Yeah, we're not even looking at that yet. Right now, we're not even looking at multiple nucleotides. At this very moment, we're just looking at a single nucleotide. Yes, what you just asked is very important, and will come up later. But for now, are there any other questions about the nitrogenous base? All right, so again, remember, it's the nitrogenous base that defines what nucleotide it is. Really quickly, the sugar, I'm going to put an X through all this, so I'm certainly not going to ask you any of these questions. The sugar has 5 carbons. 4 of them are in a ring. 1 of them is above the ring. The ring also includes an oxygen atom. If we're talking, I don't want to dig too deep into that. The carbons are numbered clockwise, so that way when scientists are talking about it, they can say it's the 1' carbon, or the 2', 3', 4', 5'. You don't need to know any of that, but if that's how scientists would describe it. If they're trying to say, if they're studying it and trying to communicate with another scientist, that's how they would describe it. You don't need to know any of that. Just know that sugar is one of the 3 parts of a nucleotide. Likewise, the phosphate group, same thing. I'm not going to ask you any of this. but I'll go ahead and mention it very quickly. Phosphorus is the phosphate group has a phosphorus in the middle. It's the phosphate part. It's acidic as opposed to neutral or basic, hence the acid in nucleic acid. And it's negative. It has a -1 charge. I'm not going to ask you any of that. For the exam, you need to know that phosphate group is one of the 3 parts of the nucleotide. And again, you need to know that between nucleotides, it's the sugar that's covalently bonded to the phosphate. So your next word for attendance, last word for attendance, other than the question that you have to ask me, is of these 3 parts, the nitrogenous base, the sugar, or the phosphate group, which one of those 3 parts defines the nucleotide? So if I say this is an A and not a T, or a C and not a G, or whatever it is, which of the 3 parts is responsible for that? Is it the nitrogenous base, the sugar, or the phosphate? Which of the 3 parts of the nucleotide, which is the monomer, right? Which of the 3 parts defines what nucleotide it is? And that's it. I'll be in my office for office hours and online if you need me, and I'll see you in lab if you have to come. I'll also be keeping an eye on my email, so if you happen to finish the lab between now and then, hopefully I'll be able to look it over really quickly and say, yeah, you don't need to come to lab. That's good. Speaking of which, the second part of the lab is to watch a whole documentary. It's like an hour and something long. So when I say we're coming to meet in lab, it's not for the documentary. It's only for part 1. You can do part 2 anytime, any place, as long as before we get it done before Friday. Make sure you use your correct section number. You guys are in 10102. So when that video asks you the question, say, hey, what section number are you in? Make sure you put 101-02, because when I'm looking for the answers to grade it, sometimes I search 10102. And if I don't find your name, I'm like, oh, they didn't do it, and I give you a 0, right? So make sure you put your correct stuff. And that's it. Sorry, I took so much time. See you guys later. Guys and gals. I don't know if that's offensive.